Thank you all for signing in today. My name is Colleen Gulick, and today I'll be speaking with you on the interplay between physical activity, dietary protein, and insulin-like growth factor 1 through a series of 3 different studies. Insulin-like growth factor 1, or IGF-1, is an anabolic hormone that stimulates cell growth and division. IGF-1 is a dichotomous hormone in that the effects may be beneficial in terms of muscle growth and repair, or detrimental in terms of increased risk of several types of cancer and mortality. Let's start with the beneficial part of IGF-1. It's great for growth and development of adolescents. IGF-1 also has an influence on athletics due to its muscle growth capabilities. It has such a substantial impact that it's banned for athletes by the World Anti-Doping Agency. The negative aspect of IGF-1 involves protein intake and longevity. For years, studies have shown that a reduction in energy intake increases lifespan. So if you eat less, you live longer. However, after reanalyzing the data, it turns out that the key factor in increasing longevity is actually a reduction in protein intake. So it should be if you eat less protein, then you live longer. It's thought that IGF-1 is the key moderator for these associations. In addition, IGF-1 is linked to cancer risk. An increase in free IGF-1 increases cell turnover, mitosis, and DNA mutation rates. So it's specifically linked with breast, colon, prostate, and lung cancer. Now we're moving on to the unknown part. Protein is the macronutrient of choice for athletes and physically active individuals. From shaker bottles to bars, athletes focus on increasing their protein intake to build muscle. This begs the question as to whether the correlation we previously spoke about between increased protein intake and decreased longevity and high cancer risk still holds true with individuals who are highly physically active. Given the diverging effects of IGF-1, discerning the factors that modulate its concentration may provide insight into how to optimize the beneficial effects of IGF-1 while mitigating any deleterious outcomes. Dietary protein and physical activity are thought to be two of the factors that modulate IGF-1. Through a series of 3 studies, we have analyzed the relationships between dietary protein versus IGF-1 via a systematic review and meta-analysis, physical activity and IGF-1 via a different systematic review and meta-analysis, which resulted in a publication. And these studies were conducted to summarize the previous literature. A third study culminated this research stream with a publication that investigated cross-sectional associations between physical activity, diet, and IGF-1 concentration in the largest sample size to date, utilizing the UK BIOBANK. For today's presentation, I'll give an overview of each of the 3 studies, reveal our results, and detail how all 3 of them tie together. Let's start with the systematic review and meta-analysis of dietary protein and total IGF-1. Overall, our selection criteria cast a very broad net in order to get an idea of what all was out there. We wanted to use the results of this systematic review to identify the literature gaps and help design our remaining studies. So, as I stated, we cast a very broad net, which in hindsight made our review incredibly cumbersome, but allowed us to see the entire literature base. The age range we chose for our study was 18 to 65 years old, and we chose that because there's a steep decline in IGF-1 after 65 years, and there is also a high IGF-1 concentration in adolescents younger than 18. So the concentrations are relatively stable between 18 and 65. We focused on healthy participants, and we extracted studies that measured total IGF-1 via serum or plasma, and then analyzed it with an effect size measure so that we were able to compare both serum and plasma measures. We separated the results according to study design. This slide shows the results for the 13 intervention studies that met the selection criteria. In a later slide, I'll display the results of the cross-sectional study analysis. Our results revealed that when compared to baseline protein intake, interventions that increased dietary protein resulted in increases in IGF-1 concentration. However, there was a high amount of heterogeneity between the study groups. So in search of the source of this heterogeneity, we conducted subgroup analyses. We subgroup by study duration with a breakpoint of 10 days. So 10 days or less was one group, and longer than 10 days was another group. No significant differences were found between these two groups. However, it did reduce the heterogeneity a bit. The study groups in which the increase in dietary protein occurred for 10 days or less resulted in an increase in IGF-1 concentration and exhibited a low amount of heterogeneity. Study groups with a longer duration had a larger change in IGF-1 concentration, but we weren't able to get rid of that high amount of heterogeneity. Still in search of the source of heterogeneity, we created subgroups for the source of protein being either a soy protein or a protein other than soy. And we did this because previous epidemiological studies have highlighted that the relationship between protein and longevity may be stronger with animal protein as compared to non-animal protein. So if animal protein is more critical to longevity, then it would be reasonable to expect that animal-based proteins impact IGF-1 concentration and thus mutageneity to a larger extent than plant proteins. In addition, previous studies have demonstrated that soy protein containing phytosterols may play a role in carcinogenesis through the inhibition of carcinogen production and promotion of apoptosis of cancerous cells. However, our results revealed no significant difference between the soy and non-soy protein groups. In addition, these subgroups did not reduce the heterogeneity. The non-soy group had a heterogeneity of 80%, and the soy group had a whopping 94%. Now that we've exhausted the statistics for the intervention studies, let's shift gears a bit and look at the cross-sectional studies that met our inclusion criteria. The cross-sectional study groups demonstrated that on average, IGF-1 concentration was 0.033 nanomoles per liter higher for every single gram per day increment in dietary protein. This is indicative of a 1.8% increase in RDI protein intake for New Zealand males and a 2.2% increase in RDI protein intake for New Zealand females. So what's our takeaway message from this systematic review and meta-analysis? The most consistent and marked result from this analysis is the direction of the association across both the intervention and the cross-sectional studies, indicating that dietary protein intake is likely associated with IGF-1 concentration. Providing a more confident estimation of the magnitude of association between dietary protein and IGF-1 will help to advise those that might be concerned about a risk of cancer on how to best modulate protein intake and potentially mitigate this risk of cancer incidence. In order to do this, we'll need more well-designed, appropriately powered, randomized controlled trials. Ideally, these studies would consist of both acute and chronic measurements in order to provide more information as to both the short and long-term effects of increased protein intake. So shifting gears a bit, let's move on to the effects of exercise and IGF-1 via a systematic review and meta-analysis. For this review, we looked at the same selection criteria as the protein review and focused on healthy individuals between 18 and 65 years. We looked at the acute response of exercise with studies that analyzed IGF-1 before and within 1 hour after exercise. The studies both had a control group and studies that did not have a control group, and we separated those out for the analysis. Let's start by discussing the studies that had a control group. The results from the 14 studies that compared post-exercise IGF-1 concentration with the same time point in a control group that did not perform exercise showed that the increase resulted in an increase in IGF-1 concentration. However, there was evidence of high and significant heterogeneity. Subgroup analysis revealed a significant effect of exercise type on IGF-1 concentration. Blood flow restriction exercise and resistance exercise resulted in increases in IGF-1 concentration. Endurance exercise and sprint exercise did not significantly change IGF-1 concentration. Next, we're going to move on to the significantly larger group of studies that did not have a control group. Results from the whopping 80 studies that compared pre and post-exercise IGF-1 concentrations without a control group gave us significance right off the bat, demonstrating that exercise resulted in an increase in total IGF-1 concentration. However, there was evidence of high heterogeneity, which is a recurring theme, which is due in part to the fact that we cast such a broad net in our study selection criteria. Subgroup analysis revealed a significant effect of exercise type on IGF-1 concentration. Resistance exercise, sprint exercise, and combined sprint with resistance exercise all resulted in increases in IGF-1. Ultra endurance and endurance exercise decreased IGF-1 concentration, and exercise with blood flow restriction, or BFR, did not significantly change IGF-1 concentration. Attempts to further investigate the causes of heterogeneity included subgroup analyses based on exercise mode, so cycling, running versus skiing, team sports, and duration. We separated duration into less than 30 minutes to 2 hours, and greater than 2 hours. All of these subgroups failed to explain the source of heterogeneity. So what's our takeaway from this systematic review and meta-analysis? The most novel, although unsurprising finding was the trend for resistance exercise to increase and endurance exercise to decrease total IGF-1 concentration. This was not particularly surprising because resistance exercise frequently results in increases in muscle mass, which relies on the mTOR pathway upregulation, and IGF-1 contributes to its upregulation. Given the timing of IGF-1 measurements for all of the included studies, which is within 1 hour after exercise, the most likely mechanism of release being captured by this sampling window is the release of IGF-1 from the skeletal muscle via an autocrine and paracrine signaling. Induction of hepatic IGF-1 release would be unlikely within the sampling window. Therefore, the appearance of IGF-1 in circulation acutely after exercise is most likely governed by skeletal muscle release caused by contractions. The results of this meta-analysis provide evidence indicating that the association between exercise and IGF-1 is dependent upon the type of exercise. Our results serve to guide individuals seeking increased IGF-1 concentration for the purpose of increasing muscle mass towards resistance exercise, while people interested in lowering their IGF-1 concentration may wish to preferentially select endurance exercise. Let's move on to our final study using the UK Biobank. The UK Biobank is a prospective cohort study comprising over 500,000 individuals recruited from across the UK between 2006 and 2010. The study protocol is available online, but in brief, participants who were between 40 and 69 were identified from the National Health Service patient registers and invited to a local assessment center to participate, where they answered touchscreen questionnaires, drew blood, and did diet questionnaires. They ran them through a whole battery. We requested and received permission to use this data to look at the association between physical activity and IGF-1, the association between dietary protein and IGF-1, and any possible interactions. So after we narrowed down our participant list to those that had completed at least 2 24-hour dietary recalls, a physical activity questionnaire, and an IGF-1 blood sample, we wound up with 60,677 participants. Dietary protein was found to be positively associated with serum IGF-1 concentration. In the fully adjusted model, which you can see on the bottom left, the things that we adjusted for, each daily 1 gram higher dietary protein intake was associated with a 0.03 nanomole per liter higher serum IGF-1 concentration, which, if you remember, is extremely similar to the 0.033 estimate that we saw in our meta-analysis of protein and IGF-1. When the relationship between physical activity and serum IGF-1 concentration was assessed in the fully adjusted model, a significant and positive association was found for moderate physical activity, which is 10 to 50 excess MET hours per week. Individuals undertaking this amount of physical activity had a serum IGF-1 concentration that was 0.13 nanomoles per liter greater than the participants that completed less than 10 excess MET hours per week. However, the high category of physical activity, which completed over 50 excess MET hours per week, was not correlated with serum IGF-1 concentration. When dietary protein and physical activity were included in the same statistical model, physical activity did not change the relationship between dietary protein and IGF-1 concentration, nor did protein change the relationship between physical activity and IGF-1 concentration. So what does this all mean? Dietary protein is positively associated with IGF-1, and this association is not influenced by physical activity. Dietary protein and IGF-1 has a stronger association than physical activity in IGF-1. So to adjust circulating IGF-1, regulating dietary protein may be more pertinent than physical activity as a primary intervention. Okay, overall, the results of these studies suggest that resistance and sprint exercise types acutely increase IGF-1 concentration, while endurance exercise types acutely decrease IGF-1 concentration, and that habitual physical activity in the range of 3 hours and 20 minutes to 16 hours and 40 minutes of moderate physical activity per week is associated with a higher total IGF-1 concentration. In terms of protein, the results of the UK BIOBANK data concur with the results of the meta-analyses and provides further evidence of a positive association between dietary protein and circulating total IGF-1. And looking at the combined effect, our results serve to guide individuals seeking increased IGF-1 concentration towards resistance exercise, while others who may wish to potentially select endurance exercise to lower their IGF-1 concentration. However, the association between dietary protein and IGF-1 is stronger than that of physical activity and IGF-1. Thus, when seeking to adjust IGF-1 concentration, regulating dietary protein may be more pertinent than physical activity as a primary intervention. One such way in which to regulate the harmful effects of a diet high in dietary protein may be to perform endurance exercise. This is evidenced by our results, which found that endurance exercise decreased IGF-1. Future research may provide insight into how modifying dietary protein and/or physical activity, and thus the IGF-1 concentration, influences incidence rates of certain types of cancer or mortality. Thank you all for your time and attention, and I'll now turn it over for questions.